second part of the course by Olga Dudko from UC San Diego. The title is How Living Systems Accomplish Things on Time, and she's going to talk about how remote genes find each other, part 2. Thank you. I'll leave you with Olga.
Good afternoon. Good to see everyone. Yesterday we talked about how living things manage time, and in particular how remote gene segments find each other in a complex environment of the nucleus. And today I would like us to talk about part 2 of the story, and I thought it might be helpful to start with a summary of what we discussed yesterday. And I put this title of the slide for myself because as a reminder that I have to condense 90 minutes of discussion into one slide, and I think I managed. So we talked about a remarkable puzzle, a puzzle to which there's really no solution yet, at least there's no consensus among researchers about the solution. The puzzle is that our genome is a about 2-meter long polymer, and it's packaged in the nucleus of on the order of 10 microns in diameter. And this is a remarkable degree of compaction. And the way that DNA is packaged into the nucleus is still largely a mystery. Of course, this packaging problem is fascinating for a physicist or for a mathematician, but importantly, it is also important for biology. And it is easy to see why. The segments of DNA that are far apart along the DNA in the 3-dimensional environment of the cell nucleus may end up interacting neighbors. And the interaction may literally be a question of life or death for the cell and for the organism. On top of the problem that we don't know the folding pattern of DNA, the folding pattern is dynamic, which is why I put 4D here, 3-dimensional space plus time. And so this makes things even more complicated and, of course, more interesting. We posed a number of questions yesterday. One of the questions was about the mechanism that governs the motion of DNA inside a cell. And as physicists, what we usually mean by mechanism is not simply the identity, I shouldn't say simply, it's often is very, very hard to find the identity of the molecules, which is of course very important and interesting. But in physics, when we are searching for mechanism, usually we are looking for some general principle that is expressible in mathematical form. We asked how remote genes, remote genomic segments find each other. And we asked several questions about the time scale of their encounter. In particular, what are the parameters that set that time scale? We talked about a particular context in which these questions can be explored, the context of adaptive immune system. And the reason that these questions arise naturally, the questions about how remote genes find each other arise naturally in the context of adaptive immune system is the nature of the so-called VDJ recombination. So let's recall, the idea is that our adaptive immune system is incredibly powerful. It can generate many more different types of antibodies than there are genes in our genome. But antibodies are proteins, right? So how is it possible? This is possible because of the, thanks to the combinatorial nature of the process by which genes that result in the antibodies are assembled. These genes are assembled from gene segments through the process of VDJ recombination. VDJ recombination occurs in 2 steps. So imagine a stretch of DNA of about 3 million base pairs long. And it has so-called D regions and J regions. And it also has a large array of so-called V regions. V stands for variable. And so in the first step of the rearrangement of VDJ recombination, one of the D recombines with one of the J's to form a DJ joint. And then in the second step, one of the V's recombine with that DJ to form VDJ. so that's the gene that will give rise to a particular antigen-specific antibody. And because there are many possibilities in which any of the V's can recombine with DJ, this helps with diversifying the pool of the resulting antibodies. I told you about the experiment that was conducted by our biology collaborators to try to understand the VDJ recombination. And in this experiment, they generated genetically modified mice which carried in their genome fluorescent markers. The first experiment was a single color experiment, and so the biologists were able to insert fluorescent markers right next to DJ regions on 2 sister chromosomes. And they were able to watch in this single color experiment the interchromosomal motion of DJ relative to DJ. So these 2 dots are the visualized DJ regions in each of the nuclei of the B cells. But because this was a single color experiment, it was not yet possible to visualize directly the motion of V that is in the same chromosome as each of the DJs, right? And so that gave us, the theorists, the opportunity, the challenge, to make predictions about how V might be moving relative to DJ. In other words, the predictions about intrachromosomal motion based on the observed interchromosomal motion of DJ relative to DJ. We talked, I think, at depth about, first of all, we saw a typical output from experiments like this. A typical output is a trajectory, literally XYZ, 3-dimensional trajectory, as a function of time of each of the fluorescent dot, in other words, each of the DJ regions. And we talked about 2 quantities that serve as powerful diagnostic tools to interpret diffusive-like trajectories, to try to extract the mechanism, to try to learn something about the environment in which the motion occurs. And these quantities are mean square displacement and velocity autocorrelation function. We also discussed how, for a broad range of processes, mean square displacement, MSD, scales with time lag tau as tau to the power of alpha. When alpha is equal to 1, that's a normal classical diffusion, right, in which there's a known result that MSD scales linearly with time. But it is possible that alpha, the scaling exponent, is not equal to 1. It can be greater than 1, or it can be less than 1, which corresponds to anomalous diffusion. When alpha is greater than 1, this is superdiffusion. When alpha is less than 1, this is subdiffusion. And finally, we talked about velocity autocorrelation functions, which tell us something about correlations, if there are any, between the velocity of genomic segment at time t and velocity of the same segment time tau later. So as the 2 velocities are separated temporally, are they correlated? Do they know anything about each other? So for normal diffusion, there are no correlations, right? So the data, the main results from yesterday in terms of analyzing the genomic trajectories were this piece. This is the time averaged mean square displacement and ensemble averaged mean square displacement for the interchromosomal motion. And the colored lines, curves, are the time averaged mean square displacements extracted from the trajectory from each of the cell, and the thick gray band is the ensemble averaged mean square displacement. In other words, MSD averaged across all the cells. And so we immediately see that the slope on the log-log plot, which is what the scaling exponent is, right? The slope is significantly less than 1. Therefore, this is not a normal diffusion. This is subdiffusion. We then noticed that the mean square displacement averaged over time and mean square displacement averaged over the ensemble agree quite well with each other, and that means that the process is ergodic. We also looked at the velocity autocorrelation functions. We discussed how to calculate them. By the way, we also discussed an annoying but important technical point of decoupling the genomic motion from the rotational and translational motion of the nucleus as a whole. And so this is how the velocity autocorrelation functions look like for various values of the discretization interval delta, which we use to calculate the velocity itself. And this is a time lag tau over which we examine the correlations. And we found that regardless of how we discretize time when calculating the velocity autocorrelation functions always exhibit negative correlations. They always exhibit anti-persistent behavior, which we interpret in terms of a pushback from the environment, something you do not expect from a classical Brownian particle diffusing in perfectly homogeneous aqueous solution. But there is a pushback from the environment that is observed here. And finally, we found something remarkable. When we replotted the velocity autocorrelation functions against the time lag that is rescaled by the discretization interval delta, we found that all these curves collapse on a universal master curve. And this is a manifestation of self-similarity. In other words, the motion has similar patterns on different scales, both temporal scales and spatial scales. Again, something that, while still beautiful, it should not surprise us if we talk about normal diffusion, but this is a very strange diffusion, right? And so it is interesting that this self-similarity is still preserved. So we briefly went over the possible mechanisms. I would like us to go over this again. The goal now is to use the signatures that we extracted from the genomic trajectories, the properties of mean square displacement and velocity autocorrelations, to identify the dominant mechanism that governs the motion of DNA segments. And this is not very simple because there is a list of possible mechanisms that are known to produce subdiffusive mean square displacement. One of these mechanisms is a diffusion that would have been normal if it was not interspersed with pausing and binding events. So a Brownian particle diffuses normally, but once in a while it finds a binding partner, binds to it and pauses there, then unbinds, diffuses normally, binds again, pauses there. And so when the distribution of pausing times has long tails, then the resulting mean square displacement is characterized by subdiffusive scaling exponents. And the corresponding mathematical framework was developed by Montrol and George Weiss and others, is known as continuous time random walk. There is another mechanism that is known to produce subdiffusive mean square displacements, and this is diffusion of a Brownian particle through a space that has obstacles, and when the hard obstacles, and when these hard wall-like obstacles are present at sufficiently high concentration, then the resulting mean square displacement shows subdiffusive scaling exponent, alpha, not less than 1. The corresponding mathematical framework is known as abstracted diffusion. Yet another possibility is diffusion through obstacles that are not hard wall-like, but springy. This is diffusion in viscoelastic environment. The corresponding mathematical framework is known as fractional Langevin motion. And as I mentioned last time, there is another possibility, a very, very technical one, but still important to consider. And this is a localization error. Just the fact that you have finite resolution with which you can determine the position of genomic segments has been shown by researchers to mimic subdiffusive mean square displacement with subdiffusive scaling exponents, even though the actual motion is normal diffusion. And so when we have a list of possible candidates, this is where the signatures that we have extracted from mean square displacement and velocity autocorrelation function analysis are going to be super helpful to us to identify the mechanism. And by the signatures, I mean, well, the subdiffusive scaling exponent, the ergodicity, the negative correlations, the anti-persistent behavior of velocity autocorrelations, and self-similarity. Because in the list of the mechanisms that are listed here, and I mentioned in addition, there's only one mechanism that is consistent with those signatures, and this is diffusion in viscoelastic environment. So before we start talking in more quantitative terms, let's discuss a little bit of intuition about what does it mean, viscoelastic environment? So first of all, our genomic segments move not in a homogeneous aqueous solution. They move in the environment of the cell nucleus, which is crowded, which is filled with chromatin fibers, proteins, some other stuff which is springy. And so as our chromatin segment makes a step, it's very likely to collide with one or more of these springs, which push back, right? And this is one of the reasons for the presence of the elastic component of the response from the environment, in addition to the viscous component. And also there is another reason, another contribution to viscoelasticity, something that actually was pointed out yesterday, and it is so obvious that it is very easy to forget about it. And this is the fact that we are talking about not about the motion of isolated particles. We are talking about motion of monomers in a polymer chain. And if you're a monomer, you, whenever you make a step, you have no choice but to stretch and compress the neighboring segments just by Newton's third law, right? And so there are restoring forces that you experience, and those restoring forces inevitably provide contribution to the elastic component of the response from the environment. So just the very fact that we are talking about polymer as opposed to an isolated particle already sufficient to have the viscoelasticity as opposed to just viscous response. Now, one interesting question to ask is everything that we've extracted so far, and also what we hopefully will, the conclusions we will make later today, how specific, how unique it is for this particular system, the motion of genomic segments in this particular locus of B lymphocytes, B cells. It turns out that people tracked the motion of DNA segments in other species and found something very similar. So, for example, researchers tracked the motion of DNA, the motion of telomeres in human cells. Telomeres are the ends of the chromosomes. And look at the scaling exponent that they have found. There's a substantial error, but obviously the scaling exponent indicates a subdiffusive motion. Researchers also tracked the motion of a particular locus in yeast cell and also found subdiffusive scaling exponent alpha in the range between 0.41 and 0.47. Researchers also tracked the motion of a locus in bacteria in E. coli and found subdiffusive scaling exponent. And so this suggests that perhaps the insights that we gain from analyzing the motion of the locus that codes for antibodies are not specific to that particular system, but are applicable much more broadly. So we established the mechanism. The mechanism is diffusion in viscoelastic environment. The corresponding mathematical framework is fractional Langevin motion. Fractional Langevin motion is governed by fractional Langevin equation. Here's fractional Langevin equation. You may or may not have seen it, but you certainly can recognize this is Newton's second law, right? We have inertia term on the left, and we have net force on the genomic segments on the right. And what are the contributions to this net force? Well, first of all, there is a drag term, right? The frictional force, negative drag coefficient times something potentially scary looking. Another possible contribution, may or may not be there, is from external force, and of course there is a thermal force. Now it is interesting to compare this fractional Langevin equation with the classical ordinary Langevin equation that was introduced by Langevin as a theory of Brownian motion. And this allows us to identify the key differences. So there are two key differences. The first difference is the form of the drag term, the viscous drag term. We can see that compared to the ordinary Langevin equation, fractional Langevin equation contains this memory kernel in the drag term in the frictional force. And this memory kernel is precisely what captures the viscoelastic memory, which manifests itself as the negative correlations in the correlation function. The second essential difference is the properties of the thermal force. We know that in classical ordinary Langevin equation, this is an uncorrelated noise, right? In contrast, in the fractional Langevin case, the noise has temporal correlations. So let's look in a little bit more detail about into the functional form of the kernel, the memory kernel in the drag force term. So the memory kernel has the following form. Here's the memory kernel, and we can see that alpha is important, right? It's a scaling exponent alpha, the one that tells us about the type of diffusion, normal versus super versus sub-diffusion. We see that it really is important in governing the behavior of the memory kernel. In particular, we can see that at long times, alpha is responsible for the behavior of the memory kernel. Now, it is interesting, actually, it's a nice exercise to check what does the memory kernel reduce to for the case of normal diffusion. Can you please look at this function and try to see what happens to the memory kernel in the case of normal diffusion? So let's recall normal diffusion is alpha equal to what? To 1, right? So what happens when alpha is equal to 1? First of all, the first term disappears, it's 0. Yes, exactly. Yeah, we just reduce it to delta function, and now try to plug the delta function back into this term. We know how to integrate with delta function, and we recover the case of Langevin, ordinary Langevin equation, which is beautiful. So this is a memory, viscoelastic memory. Now the noise, the thermal force, is Gaussian distributed with 0 mean, and with the variance, which, unlike the case of ordinary Langevin equation, has temporal correlations. So there's a memory kernel here, and as you probably know, this statement is known as the fluctuation dissipation theorem, right? And so this can be viewed as a generalized fluctuation dissipation theorem. Question?
And this both terms in the frictional force and the temporal correlations for the Gaussian, for the noise are the same case there?
Of course, that's a memory kernel. K, right? You're asking about K?
Yeah, and they are the same. But
yes, of course.
Yeah, okay.
So these terms are not independent.
Okay,
They are
okay,
related.
yeah.
It's a very important point. Thank you. It's a very important point, just like here, right? They are related through the fluctuation dissipation theorem. Yes.
So you are not introducing memory twice. It's just that they appear in two places.
No. Yeah, that's the same K. Thank you for this question. Okay, so we have an equation. We can finally talk about things quantitatively. In particular, having an equation allows us to set up a tool, a simulation tool for example, for testing predictions. We still have to make those predictions, right? But let's take a look at what kind of simulation tool we have. So here's the fractional Langevin equation. I rewrote it for little r, which is the dynamics of the distance, right, between the two segments, the relative distance. And so we can imagine the following setup for simulations is a Brownian dynamic type simulations, where we have two Brownian particles, one representing the V segment, the other representing DJ segment. They diffuse in some confinement of radius capital R. They diffuse with the diffusion coefficient D. For simplicity, let's assume they all have the same D, and that D is constant. Now, the diffusion coefficient, do you think we can, rather than just making it up, we can, in our simulations, we can actually use the value that we can extract from the experiment? Can you think of the way to get D from any of the experimental outputs we've considered so far? Absolutely. Yes, you're right. So on a log-log plot, MSD, right, is a linear graph. The slope gives us alpha, and vertical offset gives us D. And so that's a great advantage, right? So we can use experimental value of the diffusion coefficient in these simulations. Now, how about the confinement R? so naively, one might say, well, the segments move inside the nucleus, so maybe R should be roughly the size of the nucleus. But as I was talking to geneticists, I learned that genomic interactions predominantly occur within so-called chromosomal domain, right? And so this chromosomal domain sets a natural size of the confinement. This chromosomal domain, geneticists sometimes call it topologically associated domain, TAD, which I think is just a fancy way of saying a bundle of chromatin loops. Okay? And so let's imagine a bundle of chromatin loops with a characteristic dimension of microns. And this bundle of chromatin loop, chromosomal domain, creates a natural confinement. We will talk about the origin of the confinement later today in much more detail, but just for us to get the idea. And so here's some segment of one such simulation. We have V and DJ moving inside a confinement of radius R. We keep track of the distance between the two segments. Whenever the distance crosses the certain threshold where we can say the segments interact, right? That's the encounter event, and so we can measure in silico measurement the time of the encounter. And of course, it's important to remind ourselves that even though what we see here are two particles moving in a sphere in a spherical confinement, these are not isolated particles moving in vacuum or something like this. These are monomers of a polymer chain. And how do we know that they're monomers? Because the way that these particles move is by us iteratively solving numerically the fractional Langevin equation, which already accounts for both the connectivity, the fact that these are monomers attached to their fellow monomers in the chain, and the equation also through the viscoelastic memory accounts for the fact that the nucleus is a complex environment filled with springy elements. We can also imagine an analogous simulation where we can introduce many, I think this is 100, say, V elements, right, and one DJ, and so we can ask a question of how long does it take, what's the time for the first encounter between any of those V's and DJ, right? So there are lots of questions we can ask, but importantly, because the simulations are based on the equation, we can ask and answer questions quantitatively. So this is a great tool for testing predictions, but now we need to make some predictions. So there are a few interesting predictions we can try to make, but so maybe I'll talk about just one of them. So this is related to the questions. How long does it take for a DNA segment to encounter its distant counterpart? And again, so we established the mechanism of motion, subdiffusive motion, right? And we are asking if that mechanism of motion is all that is there, there's nothing, else, then what is the time scale that will result from that mechanism of motion? And if we find that just by subdiffusively moving inside the nucleus, the segments manage to find each other on a reasonable time, half an hour, as opposed to days or weeks, right? Well, that is such a joy for a physicist, because then we will have found that the general physical process can accomplish this task, the task of bringing distant elements into proximity and letting them interact. But if we find that the resulting times, encounter times, are ridiculously long, that's also interesting. This will give us the basis for hypothesizing some other biologically regulated mechanisms. So the question about how long does it take for DNA segment to encounter its distant counterpart is an example of the first passage time problem. You may be familiar with this. First passage time problem is a problem that asks about the time for a stochastic variable to reach a specified value for the first time. The first passage time problems arise across biology. They arise outside of biology in physics. They arise in the behavior of stock markets, in the weather predictions. So it's a very broad formulation. And here we are interested in the particular context of the first passage time in the context of genomic segments finding each other. What I'd like us to do now is to try to make a prediction that is based on very simple, in principle at least, simple dimensional analysis. So we all are familiar with dimensional analysis, and we want to use the scaling arguments to make some predictions about the time scale. So first of all, let's start with the obvious. The dimensions of the square brackets here indicate the dimensions of the quantity that is inside the brackets. The dimensions of mean square displacement are the dimensions, the length squared, right, always. And based on the scaling that we know for the mean square displacement, it scales with tau, the time, as time to the power of alpha. We, with the coefficient of proportionality, diffusion coefficient, we can say that the dimensions of MSD can be written as the dimensions of the diffusion coefficient times the dimensions of time to the power of alpha. Okay? And from here we can express our time. So the time is the quantity with the units of length to the power of 2 over alpha times the diffusion coefficient to the power of -1 over alpha. So the time is our first passage time, or mean first passage time that we are interested in. The diffusion coefficient, well, we all know what the diffusion coefficient is, right? So it's, we got some scaling with the diffusion coefficients, -1 over alpha. But how about the length? So we are actually facing the typical problem that arises often in dimensional analysis. So, for example, every physicist could very easily prove that E is equal to mc squared, that energy must be mass times squared speed, but it may require Einstein to interpret that speed, right? Whose speed is this? The speed of light. And so here it's a similar problem. We showed that the time scales with length as length to the 2 over alpha, but the question, what length, what quantity with the dimensions of length is this? So we need to use a little bit of intuition to try to establish that quantity, the quantity that is relevant in our mean first passage time problem. So what is the quantity with the dimensions of length that might be the most important one for setting the time scale? There are several possibilities. So one obvious possibility for the quantity with the units of length is the genomic distance, the distance between the two segments along the contour length of the DNA, right, or along chromatin. Another possibility is the quantity with the units of length, which is, we can call it interaction distance. Interaction distance is the distance at which we can declare the segments to be interacting, right? So how close they have to come to each other to one another to be able to interact. Another possibility is the size of the confinement in which the segments have the freedom to move, right? So it's another possibility, another quantity with the units of length. Now, we are talking about long-range genomic interactions. Our genomic segments are millions of base pairs apart along the DNA. These are very long genomic distances, so long that our segments really do not realize that they belong to the same chain. And so in this sense, the genomic distance is not really a very relevant parameter in predicting the time scale. Interaction distance, how about interaction distance? Interaction distance is an important parameter in normal diffusion in determining the mean first passage times. But we are dealing with not a normal diffusion, with anomalous diffusion. We are dealing with sub-diffusive motion with rather small values of the scaling exponents alpha. It turns out that for sub-diffusive motion, a strongly sub-diffusive motion, the precise value of the interaction distance does not matter that much. And this is related to the fact that the diffusive trajectories for the sub-diffusive motion are, we discussed it last time, yesterday, they're really quite rugged, right? And so mathematicians call these types of random walk compact exploration walks, and they have shown that for compact exploration random walks, the interaction distance has a second order effect. It is not as important in determining the time scale of encounters between two particles or segments, or whoever walk we are examining. So these two are out. And as a result, the size of the confinement, this topological domain, bundle of chromatin loops, emerges as the quantity with the units dimensions of length that is relevant in our first passage time problem. Let's call this size, characteristic size, capital R, and we now have a prediction. Our prediction is that the mean first passage time, or in other words, the average time for the first encounter, scales with the size of the confinement as R to the 2 of alpha, and with the diffusion coefficient as D to the negative 1 of alpha. That's a very concrete predictions. Before we proceed, I think is there a question?
So I just wanted to clarify what R is, because first I thought it was sort of the radius of a sphere centered at the segment in sort of a three-dimensional radius. And then I started thinking that R is a distance along the double helix, like a linear distance along the double helix. Because like in the figure, it looks like the orange segment can interact with some distance R along the double helix, but not with some other segment that is very close to it, but far away along the line, if that makes sense. So I just wanted to clarify what R is.
So the distance along the double helix, so really we should here talk about the distance along chromatin, remember, because DNA, but yeah. So it is not really relevant here. Why? Because the two segments are so far apart along the contour lengths, right? So it really doesn't matter. As I said, they don't probably realize that they belong to the same chain. And so the natural quantity with the units of length that emerges is the size of the spatial confinement. Obviously, this is not a sphere or anything with a continuous boundary, right? It really is a neighborhood, if we can call it so, that is formed by chromatin and chromatin loops, right? And these loops are, there's so much can be said about it, but we will just not have time, but perhaps after the lecture. But chromatin loops, they can be anchored at particular points, and they also can form bundles. We'll talk a little bit about it later. And so this bundle of loops is what people who study genomic interactions, biologists, call topologically associated domains. The name, I think, was meant to suggest that the genomic the genomic regions get associated with each other within that topological, neighborhood, right? We'll talk today about it in more detail, but this is a very reasonable question. Yes?
So in general, in mean first passage time problems, I guess the initial distance is quite relevant.
For normal diffusion, it certainly is. Yes.
I would put the initial distance there, naively. But I guess the initial distance is kind of similar to the confining domain distance, right?
No.
The average initial
No.
distance?
So the confining domain is what we just talked about, right? The initial distance is the distance at, sorry, you said initial, not interaction distance. Sorry.
Initial,
right?
yes.
Yeah, initial.
They are proportional.
You are right. Yes, in that case, you are right. It is similar. Initial or average spatial distance.
Yes.
Indeed. Yes. Yes. And so in the sense we can say, well, what is it that determines the initial or spatial distance? Certainly not the genomic distance between the segments, right? Because the question is how far the two segments are initially. That depends on the three-dimensional fold of chromatin. It has little to do with how much contour length is there between the two segments. So yes. Okay, so let's look at this prediction. So first of all, why this prediction is remarkable? It wouldn't have been particularly remarkable if this was a case of normal diffusion. Normal diffusion, remember, alpha is equal to 1. But by the way, in that case, it would have to be modified because the interaction distance would have entered there. But it really is interesting for the case of subdiffusion. Why? Because subdiffusion means alpha is small. It can be really quite small. And when alpha is small, this number can be quite large, right? And in particular, when alpha is equal to 0.5, which is a scaling exponent we found for the interchromosomal motion, we still have to soon find what the analogous scaling exponent for the intrachromosomal motion. For the interchromosomal motion, it's 0.5, so we are assuming that it's at least similar for the interchromosomal motion. Well, for when alpha is 0.5, then this power is 4. And this means that if our confinement, spatial confinement, say shrinks by just a factor of 2, what happens to the mean first passage time? It decreases by a factor of 2 to the 4, which is 16. 16 is a lot, even for biology, right, where people tend to think in like orders of magnitude. This is a very strong dependence on the time scale of encounters on the spatial dimension, the size of the confinement, and it is strong specifically because the motion is subdiffusive. So we have concrete prediction, and we have a validation tool. So far, it's simulations, right? And so this is our simulation. Remember, we are iteratively solving fractional Langevin equation. This is our dj, this is our v. They move by us, solving fractional Langevin. And we can put our two segments in v and dj in a confinement of radius 1 micron. That's a reasonable approximation, for a reasonable value for a size of this chromosomal domain, and we can repeat this experiment, this simulation, many, many times to generate the distribution of first passage times, and we can find the mean first passage time, and it is, what is it? It's half an hour. That's a very, very reasonable time scale, right? We are talking about the encounter time, right? The encounter may or may not be productive. In fact, some people estimated that every fourth encounter is productive, and so with half an hour time scale, system can actually afford having only 1 out of 4 encounters being productive, so the segments actually interact and recombine and form that VDJ gene. So let's now really test the prediction. So we can repeat what we just did, but now in a confinement that is twice smaller, right? The confinement of 0.5 micron in size, generate the distribution of first passage times, and look at the mean. First of all, it's slightly over, 2 minutes, right? But remarkably, it is actually 16 times faster, just like this scaling predicts, right? I would like to stress one important thing, why this test, this agreement is very non-trivial. We obtain this by very basic dimensional analysis. We simply use scaling arguments. We made no assumption about the underlying mechanism, whether it's fractional Langevin motion or obstructed diffusion or continuous time random walk or something else, right? No model assumptions. In contrast, the simulations are based on a very concrete equation which corresponds to a very concrete model, fractional Langevin motion. So this is a true test, right? And this agreement is very encouraging and quite remarkable. We can also play with other things. For example, before we do that. So this is more than just a curious exercise. I learned that B cells do something interesting. As B cells progress from an earlier stage of development to a later stage of development, the nucleus, and as a result, the particular locus we're interested in, shrinks. It gets more compact, coincidentally by about a factor of 2. And when I shared this research at a conference where there were lots of biologists who were not immunologists, they were working on very different systems, they pointed out that many other cells, or nearly all cells actually do this. As they develop, as they progress in their development, they undergo compaction. And it was largely a mystery for biologists why do cells do this. And so what we are finding here suggests that at least one advantage of this locus compaction is, well, decreasing the size of the spatial confinement, and as the result, decreasing the mean first passage time, speeding up the time, the rate of distant genomic elements finding each other. And once again, this is possible because the underlying motion is subdiffusive. It is for subdiffusive motion and small values of scaling exponent when this sensitivity of the time scale on the spatial dimension is really important. So we can also play with other setups. For example, we can ask how long does it take for any out of, say, 100 of V segments to find a DJ segment, let's say in the same size of the confinement, and we find that the resulting time is actually a fraction of a second. And so roughly 1 over 100, 1 over n. And so there are two important conclusions. One of them is that at least from simulations, we're getting timescales that are surprisingly, astonishingly, I would say, fast, short, right? And the second conclusion is that at least based on the simulations, our scaling that suggests the strong sensitivity of the timescale on the spatial dimension of the confinement is valid. So everything, yes, question. Should I go back?
I just wanted to ask before you move forward. In the previous part, you had just with the color for the DJ, you could also do these predictions for the meeting times of 2 DJs. No, isn't it possible?
So the question is about the meeting time between 2 DJs,
yes,
right? You know, they actually never meet, or almost never meet.
Okay.
So they are on different chromosomes. Chromosomes, I'm not even sure these 2 chromosomes are in the same neighborhood of the nucleus. And it's true in general that there's very little genomic interaction. They do occur, but they are extremely rare between the segments on different chromosomes.
Okay.
Yeah.
Okay, thanks.
Just another one. In your modeling, you have to know alpha a priori, right? Because alpha enters the kernel. So you're going to simulate
yes,
the
but
dynamics.
alpha, we get it from experiment, right?
Sure.
And diffusion coefficient, as somebody already pointed out. Yeah.
Sure.
Yeah. Yeah.
Great. So you basically simulate the dynamics in the model, and you get alpha. You can get alpha back again. So it's self-consistent, right?
Yes.
Okay.
Yes.
Great.
Yes.
Implications on, let's say, the theories of origins of life, right? Where they say, okay, the
implications where again?
theories of the origins of life, where creating compartments, cells, kind of accelerated processes, and I mean maybe this could be some kind of evolutionary kind of advantage.
That's a very interesting thought. Yes. Yeah. Yes. Thank you.
Another one here.
So, how sensitive is this to initial conditions, right? Because you have the memory kernel that kind of...
By initial condition, you mean the distribution of the segments or where they initially positioned?
Yeah, right.
Yeah,
and to do the integration, you need like information from the...
Yes, it's a very good question. And what we did, we simulated basically all possible reasonably sounding conditions from random to distributing on a sphere. Yeah, and what we found is that the precise value of mean first passage time depends on that, of course, but very mildly. So we are always within that range. So that was a very important, indeed, thing to check, that we are not, the results are not unique for that particular finely tuned initial condition. I think the one that I showed you are probably random. Yeah, but we did try other configurations. Very good point, thank you. So everything we talked about now was based on single color experiments where we used the observed interchromosomal motion of 2 segments on different chromosomes to try to make predictions about the motion of V and DJ in the same chromosome. But eventually, the great breakthrough came, and it was possible to actually introduce the second color. This was done by a former postdoc in my biology collaborator lab, Nimish Khanna. So I mentioned yesterday that it was really very hard to make it work just with a single color. And one of the reasons is that the standard labeling techniques that work for E. coli or yeast or other classical models, they absolutely do not work for B cells. B cells are just not happy. And so it really required a radically different approach to make it even a single color experiment work. And so finally, it was possible to insert the second color. And now we had the opportunity to directly visualize the motion of V, not just DJ on different chromosomes. By the way, of course, it was important for us to make sure we reproduce the original single color findings. But now we could observe directly the motion of V and DJ in the same chromosome. And we had the opportunity to test our predictions, not just from simulations, but by looking at the videos. And speaking of videos, here's one such video. So the observation time was quite impressive. It was about 1 hour. And for technical reasons, each nucleus contained 3 dots, not 4. Is related to the procedure of the fluorescent labeling, that one of the green dots marked the location of DJ on one chromosome, as before, and then a pair of green and red marked the locations of DJ and V on the other chromosome. Right? So there are some, this is an example of an experimental trajectory that was measured in these experiments. So these are trajectories from 2 nuclei. These are V and DJ in the same chromosome, and this is DJ moving on the sister chromosome, and here's V and DJ, and here's another DJ. And once again, as before, the trajectories look diffusive-like, and just as we discussed, we will not repeat it. Last time, we could now use mean square displacement and velocity correlations. We are facing the same problem with translation and rotation. Here, the problem is more severe because V and DJ in the same chromosome are much closer to each other. And so we had to invent actually 2 independent procedures to decouple the motion of interest, the motion of genomic elements, from the rest. Not that the rest is not interesting, but it's not the focus of this study, right? And I will not have time to tell you about those 2 procedures, but what gave us confidence is that 2 independent procedures gave us the same error correction, and so that was encouraging. But other than that, we now could ask some additional questions. One question we could ask is about the environment. There are already several questions about the nature of the confinement, right? And so if your genomic segments, how does it feel to move through that very complex viscoelastic, right, environment of the nucleus? And also another interesting question was, so if we imagine, we are physicists, so if we imagine a phase diagram, where should we expect our genome be? Should we expect it in a solid phase, or is it a liquid, or something else, or somewhere in between, right? And I think what I find fascinating is that questions like this, questions about the phase of matter, these are physics questions. These are the questions that people have been asking in the traditional context, like, condensed matter. And I feel that we are very fortunate to be able to ask these questions now in the context that is so much more cool, I think, in the context of life. So the first observation that we made just by watching these videos was this. We found that, so imagine you have a bunch of cells under the microscope, and in all these cells, the genomic distance between V and DJ is identical, right? So there's a biochemical method of labeling a particular V and particular DJ, and so the genomic distance is the same everywhere. But the spatial distance is, of course, generally speaking, different, right? Because spatial distance between V and DJ depends on a particular folding pattern of chromatin in that particular nucleus. And so what we found is that there was a subset of cells in which V and DJ, red and green in the same chromosome, were initially, at the start of imaging, were relatively far from each other. By relatively far, I mean 600 nanometers and more. And what's interesting is that throughout the entire imaging time, they remained separated. It was obvious that they were moving relative to each other, but this motion was like this. They would really almost never approach each other to the point where we could say they interact, and they also didn't really diffuse away. So they were fluctuating, and so that the distance between them was fluctuating, but around an average value, which itself was not changing much. And then there was another subset of cells where V and DJ, green and red, again, the genomic distance is the same everywhere, but spatially they were in proximity, spatial proximity at the start of imaging, and they remain largely in spatial proximity throughout the entire imaging time. So again, they were fluctuating, but they would really not diffuse away. And so that suggested that perhaps there are some chromatin configurations that trap our elements, relatively stable, flexible, but relatively stable chromatin configurations that trap our segments at a essentially constant average separation. Now, we can make this observation quantitative by taking the distance, spatial distance. Again, genomic distance is the same for everyone, but spatial distance depends on a particular fold or chromatin. And in a given nucleus, taking the spatial distance and plotting it as a function of time for each cell. And we can also color code these distance trajectories based on, let's say, the initial value, the initial separation or average separation. And as a result, there is this remarkable rainbow pattern which indicates, we call it the demixing effect. So the trajectories don't intersect, the distance trajectories don't intersect much, right? What we see from this rainbow pattern is that the segments that were far apart initially, they fluctuate, right? They move relative to each other, but they fluctuate around, the distance between them fluctuates around the average value that essentially remains constant. And those that were in proximity do the same. So that's a quantitative way of expressing our initial impression, right, by just looking at the videos. So let's now look at the mean square displacement, velocity autocorrelation functions, and try to understand what's going on with V and DJ. Yes, yes.
Does that mean distance compared to the maximum distance that the genomic distance, so that you could find out the maximum distance they could be separated, like by stretching?
We can see the maximum distance right from the distance trajectories, right? So I think your question then about the fluctuation, the extent of fluctuations about the average value.
Yeah, I mean, but compared to the genomic, given the genomic distance, then you have a spatial maximum distance?
Yeah, so it's nowhere near
okay.
that. Well, let's also recall the genomic distance. We can, I guess, especially if we had a Blackboard, we could convert mega base pairs into units like nanometers, centimeters, meters, right? And so, yeah, so this is not even close. Yeah, it's an interesting question, right? So really, DNA, they never get as far as they would be in a essentially extended configuration. So let's look at the mean square displacement. Just as before, we are looking at the radial mean square displacement, remember, to get rid of rotational translation. And these are the results. So this is the mean square displacement and sample averaged. The top curve shown in green is the result that is meant to confirm, and it does confirm, thankfully, what we already saw in the original single color measurement. This is the mean square displacement of the relative motion of the two DJs on different chromosomes. And we see that it is indeed subdiffusive, and the scaling exponent starts out at 0.7, but then goes down to 0.5 and 0.4, very similar to what we got from the single color experiment. But what's really new and exciting is this. This is mean square displacement for the relative motion between V and DJ in the same chromosome. It is subdiffusive, as we have assumed, right? But what perhaps is surprising is that the scaling exponent, it starts out at 0.3, but then goes down to 0.2, even 0.1 at longer time scales. So the motion is not just subdiffusive, it is strongly subdiffusive for the intrachromosomal dynamics. Oh, I wanted also quickly mention that these faint lines are the MSD before the error correction. I told you about those two procedures we came up with to correct for the error. And so the error is not huge, but we can see that it is also not negligible at short timescales, over short timescales. Now, okay, so the motion is strongly subdiffusive. What can we say about the confinement? Well, we can gain some insights by looking into the velocity autocorrelation functions. Just defined as before velocity autocorrelation functions, but now we are talking about velocity autocorrelation for the intrachromosomal motion. Just as before, velocity autocorrelation functions exhibit negative correlations, right, which is an indication of pushback from the environment. But look at the values at the deep. The values at the deep for all the values of the discretization interval delta are very close to negative 0.5. What's special about negative 0.5 is that it is a theoretical limit of so-called extreme confinement. So we find that the confinement, the environment in which the element move is springy, right? There is pushback, but it also is really quite strongly confining. And now let's do the same thing we tried with the interchromosomal motion, where we took these velocity autocorrelation curves and plotted them against the rescaled time lag, tau over delta. I hope that you remember that tau obviously is the correlation time, right? But delta was the time scale, the time interval which we use to calculate the velocity itself. And so when we plot velocity autocorrelations against the time rescaled time lag, a beautiful collapse happens, just as before, even better, perhaps, right? And so we found that, yes, self-similarity is still there. In fact, as you get used to this kind of data, you could have predicted the collapse just from the pattern of these curves, because the value of the deep is very consistent. And so what we conclude from the velocity autocorrelation analysis is that the environment in which our segments move exerts a pushback, and that the resulting motion has similar patterns on different scales. We also found that the motion of the same locus, same chromosome segments approaches the limit of extreme confinement. So we can now see if we can validate our theoretical predictions. We successfully validated them with simulations, but now we have a real test, because we have the direct way of extracting what's happening between B and J directly from the experiment. So remember, our first prediction was that the encounter times were remarkably short, in the range between second, even a fraction of a second, to just a few hours. And this is despite this seemingly inefficient mechanism of motion, subdiffusive random walk. And we can now take the subset of cells where V and DJ were initially in spatial proximity, and we can look at those trajectories for the distance as a function of time, spatial distance between V and DJ as a function of time. These are a few representative trajectories from such cells. And whenever the distance crosses a threshold is the value of the distance which we can with confidence say, well, it's a potential encounter, right? The corresponding time is the first passage time. And we found that for all the cells, the first passage times were within the range of seconds to an hour. So it is quite remarkable. Subdiffusion is not as inefficient after all. And our second prediction was even more detailed. This was a prediction about the scaling of the first encounter time with the size of the confinement. And remember, there's this interesting property of subdiffusive random walk, that subdiffusive motion, that because alpha is small, the dependence of the time on R, on the size of confinement, is very strong. So we can take from these distance versus time trajectories the values for the first passage times, and we can plot these first passage times as a function of, well, the function of R. And now the question is, what is R? And I guess you actually mentioned earlier that R is analogous to probably a good proxy for the size of the confinement is the spatial, initial, or average spatial distance between the segments. And so what we can do is we can plot all these first passage times as a function of, say, initial or average spatial distance between the segments. And we can do it on a log-log plot, which is often illuminating. And this is what we get. So these data points for the first passage time are from these trajectories, and they're plotted against the corresponding mean spatial distance. And what can we say about the slope? So let's look again at this prediction. If we take log of both sides, then the slope is predicted to be 2 over alpha, right? Now, we saw from the plots of mean square displacement, here repeated again, the value of alpha, well, it wasn't perfectly constant, but on average it was about 0.25, and therefore the predicted slope is 2 over 0.25, which is 8. And this is a line with a slope of 8. So I think it's a very good agreement. So I would like to highlight 2 important conclusions from this is that, well, we know that in general diffusion, this is actually, I find from talking to my biology colleagues that it's really surprising for them to realize that diffusion can do anything helpful, anything useful in the cell, but we know that it can be a very useful mechanism of transport on short spatial times length scales, right? Short length scales. But here we are dealing with subdiffusion, for which MSD grows very slowly with time. And it might seem hopeless at first, but what we are finding is that subdiffusion can actually ensure short mean first passage time for genomically remote DNA segments. And also important conclusion here is that it is a property of subdiffusive motion is that it is sensitive to the size of the confinement because of this particular scaling. We have not much time left, and so as the last sort of piece, I would like us to talk a little bit about the origin of the confinement. So far, we've been talking about this rather abstract, entity, confinement, size R. I mentioned something about chromosomal domain, but with the 2-color experiment, we are now in a position to hopefully say something more concrete about what creates that confinement in which the genomic segments move. So we took the antigen receptor locus, all this wealth of genomic information that we know, and by the way, what we know is that the entire locus is about 3 million base pairs. The red lines show the DNJ segments, and this is a particular DJ that was marked. The blue vertical lines show the array of V segments, so there are 200 or even more of them, and some of them are really far away from DJ. And so, and yellow lines indicate so-called CTCF binding sites, which is a whole separate story, but for the purpose of this lecture, it's just important to know that CTCF binding sites are the sites where usually chromatin loop is anchored, okay? So marked are V and DJ that were tagged in this particular experiment, and we can see that the distance between the V and DJ genomic distance is huge. It's 1.8 million base pairs. And so we took all this wealth of genomic information and, in the spirit of theoretical physics, idealize it as a bead spring polymer. Just to important thing to note, this of course should not be confused with nucleosomes in the thin fiber, chromatin fiber, right? These are not those beads. We are talking about bead spring polymer as a model for our chromatin, where each bead represents some portion of length of chromatin, and it actually is a non-trivial task about how large that bead should be. We took some time to try to understand and estimate things like persistent lengths and packing density to decide on the reasonable size of the bead. But anyway, so this is our chromatin, and so with this model we can set up molecular dynamic simulations. Now, we actually have experts here on molecular dynamic simulations, and this is really a very powerful tool. But there are also a few challenges, and so one of the challenges is the parameters at which we run the simulations. As I said, there's really little that is known, even about the persistence lengths of chromatin fiber. The reason little is known is because people don't really know how that fiber looks like. Remember, yesterday we talked about how in the textbook it's nicely pictured as a 30 nanometer thick fiber, which nevertheless there's a consensus among researchers that it simply does not exist in living cells. So anyway, we looked at very different studies using different experimental approaches, such as Hi-C that you may be familiar with, chrome EMT, electron microscopy, tomographic studies, 3D FISH, from which we extracted various estimates of parameters of chromatin, put them together, and finally we're able to convince ourselves that our bead spring polymer model of chromatin is reasonable. And so this is our immunoglobulin heavy chain locus. And another challenge of molecular dynamic simulations is that there is really no limit to how detailed and complex your simulation may be, right? So we have our chromatin, but then we can also add this protein and incorporate that protein and something else, right? But where should we stop? And also, additionally, there's really many essential molecules, many proteins that have not yet even been discovered, have not been identified, that are important for this particular locus. And so we, once again, decided to adopt a very traditional theoretical physicist approach, where we start with, well, maybe not quite a spherical cow, but something that is really a minimalist model. And with this model, we ask ourselves, can we reproduce an experimental observation? And to make our life even really harder than it already is, we insisted that our minimalist model should be able to reproduce multiple independent sets of experimental data measured by different people using different techniques, right? And if it cannot reproduce those data simultaneously, only then we are justified in increasing the complexity of our model. So if you're familiar with cooperativity and oxygen hemoglobin binding, this is essentially how the model, model of cooperativity was made. Starting from something very simple and then iteratively going between theory and observations until you reached the level of complexity that is just enough to explain experimental observations. Anyway, so here we have our polymer, and we have several experimental observations that we want our model of our locus to reproduce. Here's observation number one. This is actually a piece of data I haven't shown you yet. This is so-called FISH data, DNA FISH, fluorescence in situ hybridization. But what matters for us, this is an experiment that tells you if the two genomic segments are given genomic distance apart along the control lengths, how far apart are they in 3D space, right? So in other words, you get the relationship between microns and base pairs. And if you take a simple polymer, unstructured polymer, then obviously the further apart the two segments that you pick are, the further apart they will be in 3D space, right? So there's a monotonic dependence of the spatial distance on the genomic distance for a simple unstructured polymer. Now, let's look at these data. This is not a very exciting piece of data. What we see here is that the mean distance across the almost entire 3 million base pairs of genomic distance of the immunoglobulin heavy chain locus, the mean spatial distance is essentially constant, right? It shows a plateau, which is something we want to reproduce with our MD simulation setup. So we take a hierarchy of polymer configurations. We start, just for pedagogical reasons, we start with a simple unstructured chain, and then we have the chain to form a loop. Then we have the chain to form two loops. And the reason 1 and 2 loops are interesting, there are several reasons, but one of them is it's also fully analytically tractable configurations. And finally, we make it form a bundle of loops. And so for each of these configurations, we run molecular dynamic simulations, and for each of the configurations, we measure the spatial distance between the genomic distance. So we start with unstructured polymer, and as expected, it's a monotonically increasing dependence, right? So this thing is simply because we have to choose a point from which we start measuring distance, just the question of the origin. But other than that, it's a monotonically increasing curve, just as expected. Now, if we have our locus form one loop, well, this is the red curve. It brings the spatial distance down. The two loops do even better, right? That bring them down. The spatial distance is down, but not quite to the level of the plateau. It really is necessary for our locus to form a bundle of loops, multiple loops, to reproduce the plateau and the actual value, what is it? It's about 0.4, right, microns of the spatial distance. And so we can make a little celebration here. So our minimalist model of chromatin of the immunoglobulin heavy chain locus forming a bundle of loop configuration is able to reproduce at least one piece, the structural data of one piece of experimental observations. And from here we conclude that at least on the global scale, the confinement that we were talking about so much comes from chromatin loops, chromatin form loops, and that naturally creates a confinement. And also, I can't resist to point out that for the specific context of generating antibodies, this really makes particular sense, because if your locus is organized as a bundle of loops, then all those V segments that are spread over millions of base pairs genomically, they are brought into roughly the same spatial proximity to DJ, right? And so this eliminates the genomic bias in which only those V's that are genomically proximal to DJ would predominantly interact with DJ. So this is good. Our confinement, at least on the global scale, comes from chromatin loops, and this is our minimalist model so far of the locus. Now let's see if our minimalist model can reproduce another piece of experimental data. So these are structural data. Let's now look at the dynamic data. The data that we have, I already showed you at the beginning of today's lecture, the data that show this remarkable demixing effect of the distance trajectories when you plot distance trajectories versus time and color code them, right? So with our bundle of loop configurations, can we reproduce this demixing effect? The answer is no. Instead, we get this mess where trajectories intersect quite a lot, right? So this tells us that we do need a global confinement as chromatin in the form of chromatin loops, but it is not sufficient for reproducing the dynamic data, because it looks like there is something that is holding the genomic elements in particular configurations on a more local scale. So there is some source of local confinement in addition to the loops. What might that local confinement be? So when we came back to our biology friends with these findings, they realized that there is a whole bulk of literature on epigenetic modifications, and just as other parts of the genome, immunoglobulin heavy chain locus undergoes epigenetic modifications, which are chemical modifications of chromatin. And one of the consequences of epigenetic modifications is that the sites on chromatin that are epigenetically modified, they acquire the propensity to cross-link. So there is some kind of bridge, we don't know yet what that bridge is, molecular bridge formed between those two epigenetically modified sites on chromatin. Okay? And so inspired by this new information, we introduced another level of complexity in our simulations, and on top of the bundle of chromatin loops, we introduced a small, very small, 5% or so fraction of cross-linkable sites with, just like shown schematically here, cross-linkable sites with tunable kinetics. And we repeated our simulation, and this is what we found. We found that this configuration beautifully reproduces the demixing that is seen in the experiment. So what we conclude is that there is a confinement on a global scale, chromatin loops, but these chromatin loops are cross-linked by a small amount of crosslinks, right? And this is the time to go to classical polymer physics and remind ourselves that a solution of polymer chains, free solution of polymer chains, is called a sol. Sol is a liquid. In a phase diagram, it's a liquid phase. In contrast, polymer chains that are bridged by crosslinks, that's what we just found for our locus, is a gel. Gel is a solid. It's a different phase of matter. We just found that the chromatin is a gel, right? In a phase diagram, it's in the solid phase. And then the natural question to ask, look, I see the time, and maybe if I can take a few more minutes just to wrap up. Okay, but then we should have some time for questions. For what? For questions. Obviously, yes. Well, we already had a few questions, but yes. Just maybe 3 minutes? Okay. Yeah, I'll do my best. So then the natural question to ask, okay, so if our locus is in the solid phase, how deep in that solid phase, in the gel phase, should we expect it to be? Is it, deep, deep in the solid phase, or is it close to the boundary with the liquid? Reasonable question to ask, right? So we can try to answer this question by using the same approach, by comparing our simulation results with yet another piece of experimental data. And this time, this is MSD. That's mean square displacement that we already looked at a few times today for the intrachromosomal motion. So here's the mean square displacement. And the alpha, again, it varied, but let's say on average, it's about 0.27. So what we can do in our simulations is we can simulate our cross-linked chains for different lifetime of the crosslinks. You may be actually wondering how strong or how long-lived those crosslinks are, right? So we make no assumption about how long-lived they are. What we can do is we can simulate the cross-linked chains with the lifetime, well, of zeros. That's a perfect liquid, that's Sol, right? With the time that is basically infinite, that's a perfect solid, right? And the lifetime of crosslinks somewhere in between. And for each of these lifetime for the crosslinks, we can compute from the simulations mean square displacement and see if any of them agree with the experimental one. That's the approach. And so we did that, and what we found is that the best agreement, in fact, very good agreement, between the experimental mean square displacement and the theoretical mean square displacement is achieved when the crosslinks are present, but they're very short-lived, 10 seconds only, the characteristic lifetime. And when I say short, a meaningful comparison perhaps is the lifetime of chromatin loop. It's on the order of half an hour, right? So they're very weak crosslinks. So the conclusion from this is that the locus is a gel, it's solid. However, it is poised near the phase boundary between Sol and gel. And there are, of course, other examples in biology where people have proposed that systems are poised at the phase boundary. The systems range from our brain to flocks of birds. And so it looks like we have another possibility. So with the weak gel model of the locus, we of course wanted, I'm trying to wrap up, wanted to make sure we reproduce the experimentally measured first passage times, and they are scaling with the mean spatial distance. So the circles is what I showed you previously from the experiment. The dots are from simulations. Good agreement. Furthermore, we actually were able to reproduce the distribution of first passage times. So to conclude, the picture that emerges is as follows. A characteristic property of a solid gel is its stability. A defining property of a liquid, which is Sol, is its fluidity and responsiveness. And there are advantages to be in the solid phase, there are advantages to be in the liquid phase. If you're in the solid phase, and there are two genomic segments, the interaction can be stabilized, right? And that gives them time to explore this neighborhood and find each other and interact. However, if you're in the liquid phase, that gives the locus the possibility to undergo large-scale rearrangements, which means that those two segments are not stuck with each other forever, but there are other segments that are brought into physical proximity to each other and now have the opportunity to interact. And now imagine you are right at the boundary. You're getting the best of the two worlds, right? So if you're at the boundary, it is relatively easy to cross the boundary, and so you can cross the boundary into the solid phase to stabilize your genomic interactions, because the crosslinks are so weak, they are easily dissolved, and then you cross back across the boundary into the liquid phase, and then your locus can undergo large-scale rearrangements, and some other elements are brought into physical proximity. Again, you go back, a liquid gel droplet is formed, you stabilize those new interactions, and so on. So the proximity to the Sol-gel phase boundary means that the chromatin has a propensity to switch between solid and liquid phases, and this gives it the trade-off between the stability, characteristic of a solid, and responsiveness, characteristic of a liquid. And we propose that this might be a mechanism that not only orchestrates antigen receptor assembly, but perhaps even more generally orchestrates gene expression. And I wanted to leave you with this this image. Being at the boundary between sol and gel means that you are, you do have some crosslinks, so you don't form a puddle on a plate, but you have just enough crosslinks to barely hold the shape. And that apparently is the criteria by which this traditional Brazilian dessert is judged. I was also surprised to know that there's actually no gelatin in the original recipe, so that makes it even more challenging. But the trick is that if you kick it with a spoon, it has to jiggle, right? But so it is just at the boundary, in the gel phase, but just at the boundary. So what we found is that genomic trajectories is a great source for extracting the mechanism of chromatin motion. By analyzing mean square displacement and velocity autocorrelation functions, we found that the motion is subdiffusive, and the specific mechanism is subdiffusion in viscoelastic environment. This mechanism remarkably results in astonishingly rapid rates of genomic associations, despite the fact that genomic elements are millions of base pairs apart along the contour lengths of DNA. The key to speed is the fact that subdiffusive motion is very efficient at searching locally because of the rugged trajectory, and also it is extremely sensitive to the spatial confinement. The smaller the scale in exponent alpha, the more sensitive the time scale is on the to the spatial confinement. What is a spatial confinement? We found that there are two levels of spatial confinement. One on the global scale comes from chromatin forming loops, and the other one on a more local scale is due to crosslinks. We found, again, by iteratively comparing experiment and theory and experiment and simulations, that the crosslinks are extremely short-lived, and that puts our genome right at the phase boundary between sol and gel, with all the associated advantages, such as the trade-off between the stability and responsiveness. Thank you very much.
So we are running a little late, so maybe there is room for one or two questions.